that ends up getting copied a couple of extra times. And over time, in the population, we've gotten places where some people do a lot of these little stutters, other people maybe don't do very many. Those are things we can focus on to look at differences between individuals. So these short tandem repeats are the things that we look at. That's where you'll have 4 or so bases repeated a variable number of times. So you'll have some people where maybe the short tandem repeat at that spot of the chromosome just has 5 repeats. Someone else maybe has 20 repeats. Well, that makes smaller or bigger DNA molecules, and we can focus on those. Make many copies, and somebody will have a big, heavy STR region. Someone else will have a short, light one. We can run those through the gels and tell them apart. Those are what we use for modern DNA profiling. This STR analysis, that is looking at how long these short tandem repeat sections are, and we have certain specific ones that are identified in the genome, and they are used in clone fighting. Typically, we're looking at 13 different sites within the human genome that have these STRs in them. And that's what you use, say, with the FBI. If they say you have a crime lab has collected some DNA from the crime scene, maybe there's been a rape case, and they want to see if the offender might already be someone in the system. That's the guy they go look at then, right? What we have are 13 different locations on our different chromosomes, known by these identifiers. Nothing is very simple there. And then you get over here to the X and Y, and there's identifiers on those too. And these are basically used to identify X and Y chromosomes. You can imagine if all we find is a fragment of bone, we may not know if it's from a man or a woman, but if there is DNA that's recoverable, we can identify whether it's male or female based on those, but then figure out who it is. That's where these 13 come in. And you can imagine that at one of those sites, like right here, you may have something where you've got a continuous repeating section of these AGATs, AG18, repeated there 7 times. If we go and compare, say, DNA from a crime scene to DNA from the suspect, we may find that these match. So we're looking, say, at this chromosome, and we say, hey, looks like our guy that we're looking at for the crime may have left that DNA. But then we look at a second one that's maybe on the next chromosome. Yeah, let's say there, the crime scene, yes, crime scene has 8 repeats there, but the suspect actually has about 15. And in that case, we'd say, well, it matched there, but it didn't match there. So, close but no cigar. We would want all of these to match to be able to say with confidence the same person left this DNA as left it at the crime scene. So what you do are a lot of different profiles for all of these. You'll have these 13 different regions that we check out. Each one of them made so that we have primers that we know will attach to either end of this section. We've already researched that. So you have a primer there, and you can run this DNA through the PCR process and make many copies of that, and then run them through a DNA profile and see if they match. So from the previous figure, this is what we would see. The crime scene, we would get these 2 fragments showing up, but then from the suspect we're looking at, one would match, one would not. So that guy's in the clear. Using the hardware, of course, you've already worked with. And this is approximately what we are supposed to be seeing in our lab. I've never seen them coming out looking as good as this, but that's mostly what happens with our lab. Here's a real case that was dealt with using the DNA technology. You had DNA from a victim. Victim was last seen leaving a bar at 2 in the morning with some guy. And Police are trying to figure out who that guy might be. They have the victim's DNA, but they don't know who she left with. They don't know who she was last seen with. Well, police ask a lot of questions, like they will always do, and eventually somebody says, hey, I think she might have left this guy Joe. I don't know his last name. Well, they look into that more, and they find a guy who matched the description. They think it's Joe. They go over to talk with him, and Joe admits knowing, say, Sally. He admits knowing her, but he doesn't know anything about what happened to her. It's real sad. Has no idea that he really doesn't have any more information than that. The cops aren't convinced. They ask if they can search his place. They go, sure, I got nothing to hide. Go ahead. They go through his closet. They find clothes that have some old brownish stains on them. They think it might be blood. They ask him about that. Say, hey, those look like blood stains. How'd that happen? And he says, yeah, well, yeah, it's blood, but it's my blood. You know, I cut myself back a few months ago and I got some blood on my pants. So it's just me. They take the clothes anyway into the crime lab. They check it out. Here we have the defendant, his DNA right there. And then here we have on the jeans, bands, kind of faint. And then here on the shirt, bands that are showing up crystal clear. Do they match him or match her? Match her. Joe goes to jail. So that was more or less a real case, because I think this is a real profile from a criminal case. CODIS, you can look this up yourself online. FBI has a lot of information on this. So if some local area, like here, has a particular crime case, and they want to submit it to this national database, let's say they want to see if DNA from a crime that they're researching matches somebody who might already be in the system, this is where they would send it to. And here's a guy where they certainly matched it with. Anybody know him?
Yeah.
Oh, not.
Ridgeway.
Yeah, Ridgeway, Barry Ridgeway. Green River Killer. This guy was one of the worst of the worst. He killed about 50 women in this area. You know, up and down Pacific Highway out here, I'm sure right in the area of the campus. This is what he was doing. He was working at a truck factory, and then in his spare time, he drove around killing women. He did that a lot. And so he actually managed to go uncaptured for about 20 years after his crime spree started. He was extremely good at getting away with it, and they didn't have the DNA technology yet. Police were always interested in him. They always thought this guy really fits our ideas of who the killer might be. He was always about one of their top 3 suspects, but the cops tried and tried and tried to crack this case, and they couldn't get it solved. And so he was walking free for a couple of decades. And when we got this new DNA technology, the King County Crime Lab came in with their #1 cold case, and this was it. They had some DNA that they recovered from the crime scene that they thought was from the killer, stuff like semen samples. They had kept all that, hoping they could eventually use it. They tested it against him, and it matched. They brought him in for an interview, one of many he'd already had. Cops had been questioning him for a long time, but he always lied his way through the interviews and got away with it. He came in for this last interview, and I'm sure he thought he was just going to lie his way through that, but they showed him the new evidence that his DNA matched the crime scene, and then he cracked and confessed and started leading them to other bodies they hadn't found yet. So he was definitely the guy, and he's put away now for eternity. There are other kinds of cases, though. Here's one locally again. This girl, Mia Zapata, she was killed on Capitol Hill in Seattle. She was a rock singer there and had left a nightclub after doing a performance and was raped and murdered on the way home. The perpetrator left almost no evidence, and cops didn't know who they were looking for, but the coroner was very forward thinking. He found a little bit of dried saliva on her that he thought was from the killer, And even though he couldn't do anything then with the saliva, he knew this DNA technology was coming, so he saved a sample. And it was several years later they were able to run that sample. At first, they didn't match it to anyone, but they checked it again a couple of years after that, and it matched a guy who was now in the system. And that guy was actually living in Florida when they did the check. That's pretty far from Seattle. But they looked at his background, a guy named Jesus Mesquia. At the time of the murder, he was living 2 blocks away from her. It was him, and he's rotting in prison now for life. And I really have to admire that coroner planning ahead, thinking, I can't do anything with this yet, but this new technology is coming, and I'm going to save this. And he did, and that's how they cracked the case. But we also exonerate people. We also save innocent people who never should have been in prison. Here's one who served 20 years for a murder that he didn't commit, finally freed. And there were 2 brothers in New York, both convicted of a murder back in 1983, and recently released in 2014, when analysis of DNA showed that they weren't the killers. And here they were, it's just ruining a person's life. They sat in prison for the majority of their lives, and it's a horrible thing to think about. The DNA technology really gives us an important tool, and we're able to convict guilty people who deserve it, and free innocent people who never should have been in prison in the first place. I'm going to show you just a short film looking at the very first instance of this being done in a crime case. Let's look at this, about 5 minutes, and then I'll give you a minute for a short break, and then we'll do the test. But this was first done in Britain back in the 80s. That was where the first instance of fighting crimes with DNA was ever done.
25 years ago, a double murder case had Leicestershire police stumped. 15-year-old Dawn Ashworth had been raped and murdered in the village of Enderby. The police believed it was linked to Linda Mann's murder 3 years earlier.
A young local man had confessed to Dawn's murder, but he had no link to the earlier case, and a confession alone was not enough to secure a conviction.
Police needed corroborating evidence, but after months of painstaking investigation, they'd drawn a blank.
In the end, it wasn't traditional detective work that identified Dawn's killer. A new scientific technique gave them their breakthrough. A new technique which would change the way we fight crime forever.
It would come to be known as DNA fingerprinting or profiling. It was the brainchild of Alec Jeffries, who at the time of the investigation was researching how DNA, the molecule that makes up our genes, varies from one person to another.
In 1984, we discovered a way of getting at really variable bits of human DNA, and that provided, totally by accident, the technology of DNA fingerprinting, and opened up this fantastic door on the world of human DNA identification.
DNA is a long string of code. Jeffries found a way to identify parts of the code that are unique to an individual. He used X-rays to visualise the results.
And this is it. These are DNA fingerprints from the very first case in the world ever to be tackled at DNA level.
On this X-ray, each column represents one person's DNA. It's their DNA fingerprint. None of the columns match completely, showing the samples have been taken from different individuals.
When I first came up with this technology back in 1984, I said, well, maybe we can use these for identification. And at that point, about half the audience fell over laughing. They thought I completely lost the plot.
Initially, Jeffries' breakthrough was used to resolve paternity and immigration cases. Its crime-solving potential was spotted by David Baker, the man leading the Leicestershire murder investigation. He recruited Jeffries and sent him DNA samples from both cases, along with that of Richard Buckland, the man who'd confessed to one of the murders.
What it clearly established was that Buckland wasn't the guilty party.
And what did that mean to you? Did it throw you, or was that what you expected?
It wasn't what we expected. You know, you can just imagine there was quite a lot of heart searching when we thought we had got a confession from Buckland. So, I mean, our first task was to get him released from prison.
This was an important result, but it left the case without a single suspect. There was no such thing as a DNA database in the 1980s, so in another pioneering move, the police carried out the first mass DNA screening. All local males between 16 and 35 were called upon to give a sample.
We'd sent them a letter inviting them to come in and asked them to bring some form of identification with them so we could check out A, the invitation, B, the identification, and then we'd take the blood sample.
Then police got a lucky break.
In this pub, a local was overheard bragging that a man called Colin Pitchfork had paid him to take his ID and give a sample on his behalf.
Police arrested Colin Pitchfork. His DNA sample was taken and they got a result.
Pitchfork's unique DNA profile matched that taken from the murder scene. It proved beyond all doubt that Colin Pitchfork was the culprit.
And he became the first man to be convicted on the basis of DNA evidence.
You pretty much single-handedly changed worldwide policing. You must be so proud of that.
Well, I'm proud, but on the other hand, if I hadn't have done this, I'm just a lucky guy that happened to be in the right place at the right time with the right idea.
Since the conviction of Colin Pitchfork, major cases like the Sower murders and the abduction of Shannon Matthews have been solved with the help of DNA. And millions of DNA profiles have been created to settle paternity and immigration disputes, as well as to identify victims of war.
But as a former police officer, I know the impact that DNA profiling and the National DNA Database has on fighting crime. It's now a frontline tool relied on by police all over the world.
The Professor Jeffries that we're talking with there, he's now Sir Jeffries, Sir Professor Jeffries, I think is how they call him, knighted basically because of the outstanding importance of this work. You notice that was the first ever DNA conviction of someone, but it was also the first ever exoneration. Police had the wrong guy. He had given a false confession. A lot of people do when they're in police custody, and this sort of thing proves it. So we had, in that case, a guy who was probably serving a life sentence who shouldn't have been, and then they got the guy who really had been. So the beginning of a new world in terms of how we're able to solve crimes, and it allows us to just recover almost a microscopic speck of blood. It's too small to even see by the naked eye, and we're able to make billions of copies of targeted sections of it, and then analyze those, thanks to the technology we're looking at here. Jeffries didn't use PCR, that wasn't available then, but he had a lot of DNA available. Okay, so I'm going to come around and pass out these tests. So we should be getting our notes and stuff.